welcome to Fast News. We've just gotten a case of another fight happening over at the pub in Brisbane. Mr. Richards reacted violently towards a stranger who had accidentally bumped into him and was left stunned by his aggression. Mr. Richards was formerly employed in the travel industry and has been unemployed since COVID-19 caused major layoffs. According to friends, he has been highly stressed, desperately looking for a job, and was enjoying a rare night out with his mates when this incident occurred. This case isn't just about a man losing his temper, it's more about why this sort of behavior seems to be occurring. This is why we'll be calling up 3 specialists and questioning them about the complex biological processes happening in his brain at that moment that may have shaped his behavior. What is it about our brain having more control over whether we stay calm or explode in anger than we ourselves seem to? Let's ask our first specialist, Dr. Fred Steane-Bear. He focused on the brain's impact on actions a second to minute before they occur. What was going on in his brain?
Thank you. Well, in that brief time between being bumped and making the shove, neurons in Mr. Richards' amygdala were probably highly excited. The amygdala is the brain center for anxiety, aggression, and fear, and it processes sensory signals rapidly, so it trades accuracy for processing speed. This low accuracy, high speed system probably underlies Mr. Richards' aggressive behavior. The amygdala just doesn't have time to consider, did he mean to bump into me, or was it just an accident, before it gives the green light to shove.
Well, we don't all go around shoving people. Which part of the brain prevents this kind of aggression?
Well, that's the role of the prefrontal cortex. It keeps the amygdala in check. It's the brain region that lets us plan and problem solve. Typically, what you see is the prefrontal cortex says, he didn't mean to bump me, it was probably just an accident. But for Mr. Richards, the neurons of his prefrontal cortex are probably hampered by his stress levels. Researchers show that in rodents, stress hormones impair communication between prefrontal neurons. Here, the receptors for glutamate, an excitatory neurotransmitter, are targeted by the body and degraded. Unsurprisingly, stressed rats show cognitive deficits. Their prefrontal cortex just isn't keeping up with the rest of the brain. In fact, Mr. Richards' aggression can be further explained by the very structure of his brain regions. It is likely that in the seconds preceding his shove, neurons of his amygdala were not only highly sensitive, but also branched and interconnected more intricately than someone not stressed out. Stress increases expression of a protein called brain-derived neurotrophic factor, or BDNF, in rat amygdalas. BDNF allows brain structures to rewire and interconnect, and by the same token, BDNF expression decreases in the hippocampus, the center for learning and memory. Put this together, and you see behaviors that are aggressive and unconsidered.
And what about Mr. Richards drinking that night? Did that play a role?
Well, given Mr. Richards' stress-induced neuronal changes, alcohol was likely a key player in his aggression. In a similar way to stress decreasing excitation in the frontal cortex, alcohol increases inhibition in the frontal cortex. After drinking, the number of receptors for GABA, an inhibitory neurotransmitter, increases in the prefrontal cortex, making it more difficult for prefrontal neurons to reach the threshold for action potential. That's why drinking decreases frontal influence on behavior. In fact, fMRI scans show that acute alcohol exposure decreases connectivity between the amygdala and a frontal region called the OFC. Usually the OFC keeps the amygdala in check with inhibitory neurons, and when there's problems with this OFC-amygdala connection, you see aggression, emotional dysregulation disorders, and particular sensitivity to angry faces. As you might expect, rats with less active OFCs and hyperactive amygdalas show high rates of aggression. For Mr. Richards, damping the OFC's control over his already sensitive amygdala was always going to increase his risk of aggression. So taken together, these studies suggest that even in the brief time between being bumped and shoving the stranger, Mr. Richards' neurons were biased towards a more rapid, aggressive, fear-based, and cognitively shallow response.
Well, thank you so much for that striving conversation. But what about hours to days before this event occurred? Let's ask our next specialist, Dr. Tarik. Doctor, tell us what kind of hormones would we be looking at in case of Mr. Richards here?
Thank you, Muskan. In recent years, scientists have uncovered compelling evidence linking hormones to aggressive behavior. Key hormones such as cortisol and testosterone have been identified as significant contributors to aggressive tendencies, though their effects can vary based on individual differences and environmental changes. For poor old Mr. Richards here, we know he's been experiencing stressful events in his life, such as losing his job, which may be causing all sorts of hormonal imbalances.
And how does stress have an effect on the regulation of hormones?
Well, when you experience chronic stress, the HPA axis activates and releases a hormone called cortisol via the adrenal glands. This is the primary stress hormone that helps control metabolism and blood pressure, while also restoring homeostasis back to its original state. This temporarily regulates stress by enhancing cognition in the short term. However, the negative long-term effects outweigh the benefits. Prolonged HPA axis activity results in a weakened immune system, significant weight gain, and most importantly, mood changes.
Could one of these mood changes include aggressive behavior, similar to the behavior of Mr. Richards?
Well, a study conducted by Bonk et al. In 2010 investigated how basal and acute HPA axis influences aggression in healthy adults who thought they were in a competition. Aggression was measured in participants using noise blasts administered by other subjects as a form of punishment. Meanwhile, HPA axis was measured using salivary samples before and after they were provoked. The study found that acute HPA axis activity was strongly correlated with aggression, particularly reactive aggression. These results are significant when explaining Mr. Richards' behaviour, given that he experienced the sudden provocation of the man bumping into him, suggesting he was experiencing acute HPA axis activity. This combined with unemployment would very likely lead to an impulsive aggressive decision. Now, the other hormone I also mentioned was testosterone. While the relationship whether testosterone directly causes aggression still remains inconclusive, we know that testosterone is associated with behaviours like assertiveness and competitive behaviour. Combined with alcohol intake, which we definitely know increases the risk of aggressive behaviour, this could be a valid explanation to what happened with Mr. Richards.
What is the relationship between alcohol and testosterone then?
Well, a study conducted by Sarkola and Erickson in 2003 investigated the acute effects of a low dose of alcohol on testosterone levels in healthy men. Each subject received a low dose of alcohol or placebo in random order, followed by blood samples to measure testosterone levels. The study found a significant increase in plasma testosterone levels following the administration of a low dose of alcohol, supporting the theory that Mr. Richards' aggressive behaviour may have been the result of elevated testosterone levels from alcohol consumption. Despite not yet confirming the amount of alcohol Mr. Richards has consumed, the study shows that even a small amount can significantly increase the production of testosterone, making this a likely prospect.
Now this was interesting. Who knew so much was happening in our bodies? Finally, Dr. Anscombe, can someone actually be genetically more likely to act aggressively?
Absolutely. See, genes are segments of DNA which contain instructions for traits, with certain variations correlated with a predisposition for aggression. For example, a study by Spicala et al. Investigated the genetic risks for trait aggression and alcohol use disorder, AUD, to predict alcohol-related aggression.
Well, that sounds technical. What information will the viewers need to understand this study?
The study calculated polygenic risk scores for trait aggression and AUD. PRS are estimates of an individual's risk for specific traits. To calculate PRS, genome-wide association studies on trait aggression and AUD, which determine correlations between gene variations and higher predispositions for traits, were used to weight individual participants' genotype based on how closely their specific gene variations correlated with the studied traits, and then sum these weighted scores to produce a final PRS. The study then tested if these scores were associated with specific instances of alcohol-related aggression. Results demonstrated that higher polygenic risk scores for trait aggression and AUD were significantly positively correlated with scores on 3 or more incidents of alcohol-related aggression. These findings ultimately suggest that people with certain gene variations, as I hypothesize Mr. Richards may have, are predisposed to alcohol use disorder and trait aggression, which leads to instances of alcohol-related violence.
Do genes also have an impact on aggression at the hormonal and cellular level?
Well, individuals with variations in genes related to serotonin, essential for mood stabilization, show a predisposition toward alcohol-related violence. Chromosome 17 contains a gene coding for serotonin transporter protein. The role of this protein is to transport serotonin from the synaptic cleft back to the neuron in a process called serotonin reuptake. Another segment of the gene, 5-HTT-LPR, codes for the length of this transporter protein and comes in 2 gene variations or alleles, being long or short. The short allele reduces the expression of the transporter protein, disrupting serotonin reuptake and the overall regulation of serotonin in the body. Since alcohol causes a significant increase in serotonin levels, individuals with the short allele can't regulate these increased levels through serotonin reuptake, destabilizing their mood when consuming alcohol. Consequently, individuals with a short allele possess a predisposition for alcohol-related aggression in the presence of environmental stresses like unemployment related to Mr. Richards' case.
You mentioned environmental stresses impacting genes. Is there a way scientists can differentiate the impact of genes versus environmental influence?
Absolutely. Take the study by Tylan et al., which utilized a twin paradigm to analyze personality similarity in read together and read apart monozygotic versus dizygotic twins to determine genetic influences versus shared and unshared environmental influences. They used a self-report personality inventory measuring 11 primary personality dimensions, including a negative emotionality, whereby high scores indicate an increased disposition towards stress and aggression. Results indicated a 61% intra-class correlation of negative emotionality between separated monozygotic twins, significantly greater than separated dizygotic twins, showing a strong genetic influence and little unshared environmental influence on aggression.
Thank you so much for that. We found out that even in the seconds preceding Mr. Richards shoving the stranger, communication and connection between his brain regions were biased towards aggression and inhibition. Then we went a bit further back in our brain to understand how cortisol and testosterone changes hours prior to the incident, which may have influenced his aggressive behavior. And then we explored how genetic variations predispose individuals to alcohol-related aggression, from influencing the expression and functionality of proteins regulating serotonin to specific genes correlated with incidents of alcohol-related violence. Thus, Mr. Richards' behavior tonight was not determined by any one factor. It's the combination of biology that extends from seconds to hours to days before the shove to the very makeup of his DNA. Hence, some of our worst behaviors are determined by our biology, but so too our best behaviors. Thank you and good night.